Hello everyone, welcome to ITLS Academy. I am Ronak Fatima and today we will discuss about vaccines for prion diseases. The disease is relatively new and having only been identified in recent years, so it means that extensive historical data and long-term studies on this progression and impact are still limited. So, does it sound interesting? Let's explore more. So, here is what we will cover. We will start with a case study on mad cow disease caused by prions. What are prions and disease caused by prions and its basic information along with pathogenesis, types, causes, and most importantly, its vaccines. Okay, so let's start with historical event caused by prions. Mad cow disease. You must have heard about it. The mad cow disease is commonly called as bovine spongiform encephalopathy, that is abbreviated as BSE, a disease of cattle that was first came into limelight in 1986 in the United Kingdom. People observed the strange behavior in their cattle. What strange behavior? Their cattle were staggering, drooling, and at one time they were behaving very aggressively, and the other time they were not even able to stand. So this left, you know, curiosity among people. So one of the cow was slaughtered, and examiners, I mean researchers and scientists, examined the brain of that cow. And what they have examined, they examined the brain of the cow was full of holes, empty spaces, due to dead nerve cells, or you can say neurons. All right, and this was because of the mysterious disorder. The cause was not known. Government assured public then that BSE is not a threat to public. However, there comes a twist. अब क्या होता है? ट्विस्ट ऐसे आता है कि after 10 years, that is, in 1996, 10 people died with similar human disorder that was named as Creutzfeldt-Jakob disease, abbreviated as CJD. All right. So what was the reason? इतना सब डर का माहौल हो गया कि एकदम सिमिलर डिजीज है, कैसे एकदम से हो गई? So researchers assumed that the disease may be caused by eating BSE infectious cows. And since then, Britain and other countries adopted strict inspection rules to keep infected meat products away from the human food supply. फिर ट्विस्ट आ जाता है, अगेन ट्विस्ट. अब क्या होता है, इतना प्रिकॉशंस लेने के बाद भी in December 2003, a confirmed case of BSE was found in the tissue of cow. Where? In the United States of America. Okay. अब क्योंकि BSE और CJD का जो कनेक्शन है, एसोसिएशन है, लिंक है, वो still it was poorly understood. So scientists were generally accepted the hypothesis that the disease is caused by proteinaceous infectious particle called prions. Okay, so what are prions? Prions are infectious protein which can cause abnormal folding in the normal protein. Okay, normal protein. कैसे प्रियन नाम पड़ा? प्रियन का नाम प्रियन क्यों पड़ा? ये नाम प्रोटीनेशियस इन्फेक्शियस पार्टिकल ही क्यों पड़ा? तो लेट्स अंडरस्टैंड क्यों हुआ ऐसा. वी ऑल नो कि microbes are the agents that cause diseases. Okay, so earlier it was thought to be virus, but we all know viruses replicate slowly within their host, um, but They have nucleic acid like DNA and RNA. However, prions does not have any nucleic acid. There is no DNA or RNA in prions, and prions are 100 times smaller than viruses. So ये तो कंफ्यूजन बहुत क्रिएट हो गई. अब तो ये तो बहुत मिक्स्ड अप हो गया. साइंटिस्ट बड़े क्यूरियस हो गए कि अब क्या हो रहा है यहाँ पे हमारे यहाँ. तो दे वर लाइक, जब उन्होंने पूरा आइडेंटिफाई किया, उन्होंने देखा ये प्रोटीन, सिर्फ प्रोटीन की वजह से ये डिजीज कॉज हो रही है. सो इसको अब क्योंकि हम लोग माइक्रोब्स तो नाम दे नहीं सकते, तो इट्स कॉल्ड पार्टिकल्स. अब ये प्रियॉन्स का फीचर ही है, इसके फीचर ही है कि ये बहुत इन्फेक्शियस है. ये नॉर्मल प्रोटीन को मिसफोल्ड करती है, दैट्स वाई उसका नाम पड़ा इन्फेक्शियस. एंड ऑब्वियसली इट इज प्रोटीन, सो उसका नाम प्रोटीनेशियस इन्फेक्शियस पार्टिकल पड़ता है. ऑलराइट, नाउ प्रियॉन करती क्या है? Prion are known to cause neurological disease in mammals, alright, like CJD in humans that we have discussed in the case study. Okay, now Stanley Ben Prisoner in 1982 have discovered prion protein, that is abnormal prion protein, and also he received Nobel Prize in 1998 for the same. Okay, now let's come to the structure. Okay, know that normal prion protein, where's my cursor? Okay, normal prion protein is abbreviated as PRPC. We will continue with PRPC. And abnormal prion protein is abbreviated as PRPSC. Here, C is, C denotes cellular or common. Okay, and here in PRPSC, C denotes Scarpi. Scarpi, the name derived from the prion disease occurring in sheep. Okay, sheep and goats both. All right. Now let's learn what are normal prion protein. Normal prion proteins are the proteins found throughout our body. Okay, in the brain, nerves, and related tissues, but especially in especially on the surface of our neurons in the brain. Okay, now how these two are different from each other, let's explore the differences. Okay, first know that normal cursor. Okay, normal prion protein structure are well defined, but this misfolded prion protein structure is polydispersed and relatively poorly defined. Okay, normal PRPC are non-infectious, PRPSC are infectious. Okay, normal prion protein are monomer and PRPSCs form aggregates. PRPC are soluble, however PRPSC are insoluble. The structure of normal prion proteins are predominantly alpha helical. Okay, let's see. Just a second. Okay. Okay. The structure of prion protein is generally alpha helical. Okay. However, the structure of misfolded prion protein is rich in beta sheets. All right. Now, we all know that if we have unwanted protein in our body, so there is an enzyme called proteases which cleaves that unwanted enzyme and digest it. Okay, similarly there is a serine protease enzyme called proteinase K. ठीक है, तो proteinase K enzyme, normal PRPC is sensitive to proteinase K enzyme. However, this misfolded abnormal PRPC is partially resistant to proteinase K enzyme. So this makes it undigestible in the body, that makes further aggregates and cause neurodegenerative diseases in mammals. Alright, now we come to our next slide. What are the diseases caused by prions? So first of all, let's learn what are prion diseases. What is the definition of prion disease? Prion diseases are a group of very rare neurodegenerative diseases that can affect humans and animals. Why very rare? Because only 300 cases per year are found in USA recently. Okay. Now, the most popular name of prion diseases is transmissible spongiform encephalopathy. All right. Just a second. All right. Uh, what is the popular name of prion disease? Transmissible spongiform encephalopathy. We also abbreviate it as TSEs. Okay. Now, the most interesting part of this disease is its incubation time. हम लोग ने अभी केस स्टडी देखा, क्या देखा उसमें? 10 साल पहले हम लोग को बीएससी का केस मिला था एंड 10 साल बाद हम लोग को सीजेडी का केस मिला इन ह्यूमंस. सो ये 10 साल का गैप बता रहा है कि ह्यूमंस में ऐसा नहीं है कि कैसे आया होगा, बीएससी के थ्रू ही हम लोगों ने अज्यूम किया, तो 10 साल का पीरियड लगा. Okay, so it shows, and other studies as well shows that there is a prolonged incubation of period of several years, prolonged incubation period of several years, but it depends on disease type. Disease type we will discuss it further. Okay, and all prion diseases are fatal because we all know neurodegenerative diseases में हमारी cells क्या होती हैं, हमारी न्यूरॉन्स हमारी या तो काम करना बंद कर देती हैं या मर जाती हैं. ऑलराइट, एंड ऑब्वियसली दे आर incurable till now. Why? We will discuss it further. Understood? Now, there are pathological alterations, especially confined to the central nervous system, and they affect structure and function of neurons. Okay, structure and function of neurons. Let us see how. Let's see how. Okay, स्ट्रक्चरल चेंज में क्या होता है हमारे यहाँ? स्ट्रक्चरल चेंज में हमारा स्पॉन्जी फॉर्म चेंज हो जाता है. ठीक है, स्पॉन्जी फॉर्म चेंज इट मीन्स when brain exhibits spongy appearance due to formation of vacuole. Okay, and alpha and amyloid plaque में क्या होता है? इसमें मिसफोल्डेड एग्रीगेट्स होते हैं जो अमालॉइड प्लाक बनाते हैं एंड इट डिसरप्ट्स the normal architecture of neuron tissue, and then there is a extensive neuronal loss, and then comes astrogliosis mean there is an increased number and size of the astrocytes, the type of glial cells. Okay, and what are the functional changes? What are the functional changes? There, we can see there is disruption in synaptic transmission and impairment, neuronal communication, and then there is in cognitive decline. There is, progressive dementia, personality change. Why? Because of the degradation in brain structure. Okay. Now, neuronal death. We all know that there is loss of neurons that leads to the decline of our brain function. Okay, and motor dysfunction. Motor dysfunction, it shows the effect on motor control, and if there is effect on motor control, what we see, the symptoms are ataxia. Ataxia means the loss of coordination. Yeah, and what are the other symptoms? It includes tremors. All right, understood? Okay, now now let's come to the pathogenesis of prion disease. See, knowing about the pathogenesis of prion disease is very important to know, to understand how vaccine works against prion diseases. Okay, so now let's come to the first picture. Now you can see this image shows the process of prion protein misfolding and aggregation, which leads to prion disease. Let us see how. First, here you can see these are normal prion proteins, okay, PRPC, okay. When PRPC interacts with foreign or infected abnormal prion proteins, they undergo conformational change. Here you can see undergo conformational change, okay. And then जब conformational change होगा तो it converts into misfolded pathogenic isoform, which is called PRPSC, okay, abnormal prion protein. And then abnormal prion protein recruits and facilitates what? The conversion of more normal prion protein molecules. So leading to the accumulation of what? Accumulation of accumulation of aggregates. Okay. Now, you can see the cycle of this abnormal prion protein of recruitment and conversion of normal prion protein continues indefinitely. Okay? And this leads to the progressive accumulation of aggregates. So, this becomes the benchmark, or you can say the hallmark of prion disease. Okay? Now, let's come to our next image. This image shows how prion protein plays role in progression of neurodegenerative disease. Let's see. Here you can see ER stress and UPR activation. Okay. ER stress and UPR activation trigger normal prion misfolding and abnormal prion protein accumulation. See this? It triggers normal prion misfolding and abnormal what? Abnormal prion protein accumulation. Abnormal prion protein sets the cascades of events. The cascades of events such as autophagy, synaptic dysfunction, dendritic loss, spongiform degeneration, and brain inflammation and neuronal death. These these this ठीक है? These pathological process initially manifested in presymptomatic phase, okay, and then this followed by early clinical phase, okay, and in early clinical phase the you know the symptom, this pathological process becomes uh, gives the mild symptoms, and later at final stage, the late clinical phase, it becomes a severe symptom. Okay. Now let's see what are the different forms of TSCs in animals and humans. In animals, we have already discussed about BSEs, and scurvy is a neurodegenerative disease that generally occurs in sheep and goat. And now CJD. CJD is the most common prion disease where there is a rapid progression of dementia and neurological decline. All right. Now there is VCJD. Now it may look similar, but these two are very different from each other. Let's see how. This CJD, the CJD, the CJD is associated with BAC and with prominent psychiatric symptoms. Okay, so this is completely linked with BSc. Okay, now here in VCJD there is additional symptom, or you can say additional problem, that is prominent psychiatric symptoms. Okay, now there are so many case studies on Kuru is the uh, prion disease that is transmitted through ritualistic cannibalism. Okay, okay. Now, fatal familial insomnia. It is a genetic form of affecting, you know, thalamus. Okay, and it generally leads to, extreme insomnia cases. Now, don't think if you have insomnia problems, so you must be, affected by prion disease. No way. That's not the only symptom. Okay. Okay. Now let's come to the next slide. Here, types of prion diseases. Basically, prion diseases are of three types: infectious, familial, and sporadic. Okay, sporadic prion diseases are the most common. Okay, why? See, इन्फेक्शस जो हमारा होता है वो इन्फेक्शंस के थ्रू फैलता है, ठीक है। वी नो कि वो इन्फेक्शंस के थ्रू फैल रहा है, ठीक है। either we consume infectious, meat, or through transfusion, or through latrogenic spread. Latrogenic कैसे कहते हैं? when accidentally or unintentionally transmit हो medical procedure के थ्रू कोई infection. Okay, एंड फेमिलियल जेनेटिक जब हमारे जीन्स में हो. ओके, बट स्पोरेडिक प्रियन डिजीज को बहुत ज्यादा सिवियर डेंजरस बनाता है ह्यूमंस के लिए. क्यों? वाई? बिकॉज, एंड ये 85% है. क्यों? क्योंकि स्पोरेडिक का मतलब ही यही है कि इट अकर्स इन पीपल विदाउट नोन रिस्क फैक्टर्स. अब तो हमें रिस्क फैक्टर्स पता ही नहीं है. जब रिस्क फैक्टर्स हमको पता नहीं है तो किसी को भी कभी भी कहीं भी हो सकता है. वी डोंट नो कि क्या रीजन थे. ओके, सो इट इज इम्पॉर्टेंट कि इसके लिए वैक्सीन हो, ट्रीटमेंट हो. ओके, सो नाउ let's come to our next slide. Here are the causes, symptoms, and complications that we have covered in every part of the slides, previous slides. Now coming to diagnosis. Now, prion diseases को कैसे diagnose किया जाए? There are several techniques like MRI and lumbar puncture, where we take fluid samples from spinal cord, blood tests, electroencephalogram, that is EEG. It is used to analyze structure and electrical activity of the brain, and through visual and neurological examination and real-time quick. These are the steps. Okay. Now, मसला कहाँ है? अब हमारी डिजीज डायग्नोस हो गई. ओके, अब क्या? अब तो हमको ट्रीटमेंट देना है, राइट? बट अभी हमने पढ़ा इन अर्लियर स्लाइड दैट प्रियन डिजीजेस आर इनक्योरेबल. अब तो मसला फंस गया, देयर इज नो ट्रीटमेंट अवेलेबल. अब क्या करें हम? यहाँ तो है, ठीक है. तो यहाँ पे हमारा एंटर करते हैं वैक्सीन. पर वाई वैक्सीन? हाँ, हम लोग को प्रिवेंट करना है ना. तो वी ऑल नो अब हमारे पास ट्रीटमेंट का कोई ऑप्शन नहीं है, क्योर का कोई ऑप्शन नहीं है. सो वी ऑल नो प्रिवेंशन इज बेटर देन क्योर. ओके, बट हाउ टू प्रिवेंट? वी नीड वैक्सीन, राइट? सो लेट्स कम, व्हाट आर वैक्सीन? ठीक है, लेट्स कवर द बेसिक्स ऑफ वैक्सीन फॉर नाउ. ओके, what are the basics of vaccines? See, vaccines vaccines are the suspension of weakened or killed or fragmented microbes or toxins or antibodies or lymphocytes that is administered primarily to prevent disease, and the administration of vaccine is called vaccination. All right, now we all know that Vaccine can provide active immunity. You can say artificial active immunity against specific harmful agent by stimulating the immune system to attack that agent. Okay. Now, vaccines are very important and very effective method to prevent infectious disease. All right. Now, let's come to the one-line history of a vaccine. The term vaccine was derived from a Latin word vacca that was used for cows, and it was derived in 1796 by Edward Jenner. By Edward Jenner. Okay. Now, we all know that there are several types of vaccines, like attenuated vaccine and inactivated or killed vaccines, subunit vaccine, DNA vaccines, synthetic peptide vaccines, toxoids, and more. Okay, so now we all know that only vaccines can be our savior. Hmm. Now, coming back to prions. First of all, why do we need vaccine? Hmm? Why do we need vaccine? Sorry about my cursor, okay? So let's come to the topic. Now, why do we need vaccines? If we, have systems to check correct protein formation and have immune system for protection. See, if protein if protein formation system are, bypassed, or you can say overwhelmed, they can hinder formation of correct protein structure, and it will definitely lead to, abnormal prion formation. But what about the immune system and what about its integrity? Immune system भी तो है हमारे पास, उसका क्या? See, immune system does not develop a bona fide immune response against the PRPSC. पर क्यों? Why? Because जो PRPSC and PRPC हैं वो एक दूसरे से आइडेंटिकल उनके दे शेयर आइडेंटिकल प्राइमरी प्रोटीन स्ट्रक्चर. ओके, दे शेयर प्राइमरी प्रोटीन स्ट्रक्चर. And since normal prions are already present in the body, the presence of either form does not trigger an immune response. Okay, so we require vaccine. But अब हम लोग को वैक्सीन तो चाहिए और वैक्सीन है ही नहीं. वैक्सीन क्यों नहीं है? Why do we lack vaccine? Government, no. See, governments are, employing several methods like containment strategies in wild years, like depopulation and selective harvesting, but they are not very effective. So we need vaccine. वैक्सीन के बिना कुछ नहीं होना. ठीक है. क्यों वैक्सीन नहीं है? These are the reasons. सबसे बड़ा reason is active vaccination is not essential. Active vaccination क्यों essential नहीं है? Because we have already learned prion diseases are rare. ये कुछ पोलियो और कोविड-19 की तरह तो है नहीं कि एक्टिव वैक्सीनेशन की जरूरत होगी, एंड इट्स रेयर, तो एक्टिव वैक्सीनेशन की जरूरत नहीं लगी तो लोगों ने वैक्सीन भी बनाना जरूरी नहीं समझी. ये होता है. एंड देयर आर सो मेनी अदर रीजंस एज वेल, जिसमें आप देख सकते हैं कि इनके पास वैक्सीन्स दे डू नॉट हैव ability to overcome the immunological tolerance, and there are so many safety concerns of inducing immune responses to self proteins. एंड क्योंकि वी ऑल नो, क्योंकि पीआरपीसी तो हमारी बॉडी में भी अवेलेबल है, वो तो कन्वर्ट कभी भी हो सकता है, राइट? तो हम लोग वो अपनी उसको अपनी नॉर्मल प्रोटीन को तो मार नहीं सकते हैं. ऑलराइट. एंड देयर आर अनसर्टेनिटीज ऑफ मैकेनिज्म ऑफ इम्यून प्रोटेक्शन. ओके, ये हम लोग आगे डिस्कस करने वाले हैं इन डिटेल. लेट अस सी हाउ. नाउ, सबसे पहले आपको ये बता दूं कि द गुड पार्ट इज हमारे यहाँ science is not about giving up. Okay, science is not giving up, so researchers are working dedicatedly on prion diseases, and there are so many ongoing research projects to develop vaccines to stop lethal impacts on humans. So now let's see how they're working and they're up to what. Okay, Now let's come to vaccine for prion disease. See, there are two approaches. Okay, scientists are working on two approaches. First, vaccines inducing self antibodies, and the second approach is vaccines interacting epitopes. Okay, so the first approach is vaccines inducing self antibodies. First of all, no. First of all, know what is इम्यून टॉलरेंस. हम लोग को ये पता होना चाहिए कि इम्यून टॉलरेंस क्या होता है. ठीक है. सी व्हेन इम्यून सेल्स क्रिएट हाईली स्पेसिफिक रिस्पॉन्स टू फॉरेन बॉडीज एंड अवॉइड रिएक्शन टू सेल्फ प्रोटीन. जब वो बाहर फॉरेन बॉडीज के लिए रिस्पॉन्स बहुत हाईली स्पेसिफिक होती है उनको रिस्पॉन्स करने के लिए, लेकिन खुद के सेल्फ प्रोटीन के लिए को रिएक्ट करने को अवॉइड करती हैं. ओके, so this is the basic definition. Okay, see, body will not produce antibody against cell protein. Okay, and since PRPC is also cell protein, we cannot have immune response against it. So we will not have any protection from both normal PRPC, normal prion protein, and abnormal prion protein. And then we will introduce vaccine in the body. So immune response is created. Okay. Okay. So now for this, where's the cursor? Okay, ignore the cursor. For this, the first method is PRP specific T cell receptor. PRPC specific T cell receptor can help. How? It will recognize PRPC bind and it will it will recognize the normal prion protein bind with it, and it secretes interleukin. See, जब हमारा नॉर्मल प्रोटीन होगा पीआरपीसी नॉर्मल प्रोटीन, वो क्या करेगा, it binds with T cell receptors and then it secretes interleukin. Okay, and similarly for PRPC vaccines with peptide loaded dendritic cells also works by secreting interleukin. जो रिएक्शन हमारा है वो secrete करेंगे interleukins, interferons and antibodies. Now सब यूनिट वैक्सीन यहाँ पे हेट्रोलॉगस वर्जन यूज करते हैं. हेट्रोलॉगस वर्जन जितनी रिसर्च हुई है प्रियन प्रोटीन पे, हेट्रोलॉगस फॉर एग्जांपल माउस से, हेट्रोलॉगस मतलब बाहर से हम एंटीबॉडी लाके इंड्यूस करा रहे हैं. ठीक है, बाहर से, अगर माइस का बाहर से माइस से ही लाके कराया तो उस पर कोई रिजल्ट नहीं मिला. ठीक है, अकॉर्डिंग टू रिसर्च टिल नाउ. ओके, बट हम अगर टू सोर्सेस से, लाइक फॉर एग्जांपल शीप एंड बोवाइन से लाके माइस में इंड्यूस करा रहे हैं एंटीबॉडी, तो वो रिजल्ट दे रही है. तो इससे हमारा इम्यून रिस्पॉन्स इंक्रीज हो रहा है. तो सब यूनिट वैक्सीन कैन बी an option. Okay, now comes peptide vaccine. Why peptide vaccines? Because this peptide vaccine, that is succinyl arginine dehydrolase, this have similar sequence like normal prion protein, so it can induce PRP specific antibody. Okay, now I hope you understood. Okay, now coming to the second approach, that is vaccines interacting with epitopes. Epitopes, first of all, what are epitopes? Epitopes are antigen determinant that are part of antigen and is recognized by our immune system, especially by antibodies, by B cells and T cells. Okay. Alright, now the second approach of vaccine development using epitopes for immune response is very helpful. How? Let's see. See, scientists are working on conserve sequence. Okay, conserve sequence of both normal. Oh God, okay, both normal and abnormal prion protein. Okay, so the conserved sequence of normal and abnormal prion protein works as antigen. Okay, now if if normal prion protein antigen induces antibodies, it binds to the cell surface located on normal prion protein and it impairs PRPSC formation. Current working of this approach is DEVA vaccine. इसमें क्या हो रहा है इसमें क्या हो रहा है इसमें पीआरपीसी एंटीजन हमारे या तो नॉर्मल प्रियन को क्या कर रहे हैं मार रहे हैं या फिर हमारे बनने नहीं दे रहे हैं पीआरपीएससी मॉलिक्यूल ओके हाउएवर यहाँ पे दूसरा अप्रोच है पीआरपीएससी स्पेसिफिक एंटीजन ये बेटर प्रेफर किया जा रहा है क्यों बिकॉज ऑफ इंक्रीज्ड सेफ्टी एंड एफिकेसी वाई क्यों इसका इंक्रीज सेफ्टी एंड एफिकेसी प्रोवाइड कर रहा है बिकॉज फर्स्ट ऑफ ऑल यहाँ पे हम लोग को श्योरिटी रहेगी कि देर इज नो क्रिएशन ऑफ ऑटो इम्यूनिटी हमें ऑटो इम्यूनिटी का कोई खतरा ही नहीं है खत्म हो गई बात यहाँ पे ऑटो इम्यूनिटी का खतरा ही नहीं है एंड द सेकेंड रीजन इज एफिकेसी वाली बात पे ये यहाँ पे हम लोग डायरेक्टली डिजीज्ड मॉलिक्यूल पे काम कर रहे हैं डिजीज्ड प्रोटीन मॉलिक्यूल पे काम कर रहे हैं किस पे पीआरपीएससी पे काम कर रहे हैं तो हमारी एफिकेसी भी बढ़ सकती है ओके तो इसके तीन बहुत पॉपुलर एग्जांपल्स हैं रिसर्च में जिसका वाई वाई आर एपिटोप वाई एम एल एपिटोप एंड रिजिड लूप एपिटोप ये बहुत कॉमन यूज हो रहे हैं आई होप यू अंडरस्टूड ओके नाउ अब आते हैं हमारा डिलीवरी मेथड पे वैक्सीन for prion diseases delivery method. Now, see, there are two methods, injections and oral. Alright, injections and oral. See, in the slide I have written oral vaccines are mostly preferred. But what about, injections? See, हमारे जो इंजेक्शंस वाले एंटीबॉडीज होंगे और म्यूकोसल एंटीबॉडीज हैं ये दोनों ही हमारे पेरिफेरल एम्प्लीफिकेशन को इंड्यूस कर रहे हैं ओके ये पेरिफेरल एक्टिविटी को इंड्यूस कर रहे हैं ओके तो यहाँ पे हमारा पेरिफेरल एम्प्लीफिकेशन हो रहा है ठीक है ये दोनों आईजीजी है ठीक है ये पेरिफेरल पे भी वर्क करता है इंजेक्शन एंड ओरल भी एंड दोनों ही दे बोथ क्रॉस ब्लड ब्रेन बैरियर एंड रीच ब्रेन सेल्स ओके तो यहाँ पे दोनों जगह काम हो रहा है ओरल हमारा इंजेक्शन हमारा दोनों पेरिफेरल एम्प्लीफिकेशन कर रहे हैं हमारे ब्लड में भी है ब्लड स्ट्रीम्स में भी एंड ब्लड ब्रेन बैरियर क्रॉस करके हमारे ब्रेन में जाके अफेक्ट दिखा रहे हैं ओके बट हियर oral vaccine has plus point क्योंकि we all know primary route of prion transmission is through mouth okay so हमारे oral में हमारा एक प्लस पॉइंट और बढ़ जाएगा हमारा ये वाला भी प्लीज लाइक oral vaccines can be beneficial and stops infection and works as you know as dushman you can say to against prion disease okay now तो हमारा oral is helpful for both peripheral and mucosal immune response and also crosses blood brain barrier and reaches brain cells however our injection method is only good for peripheral and just crosses uh, ब्लड ब्रेन बैरियर रीचेस ब्रेन यहाँ पे हमारा ये है कि बस माउथ से इन्फेक्शन तो रोक नहीं पाएगा तो फिर क्या फायदा इतना तो इफेक्टिव होगा नहीं वो जितना ओरल हो जाएगा ओके एंड द प्लस पॉइंट ये भी है कि ओरल वैक्सीनेशन का कि हम लोग कैटल को मतलब हर कैटल को वन बाय वन जाके इंजेक्ट तो करेंगे नहीं राइट सो तो इजी वे ऑफ वैक्सीनेशन है ओरल मेथड है क्यों क्योंकि डिस्ट्रीब्यूशन इजी है ओके सो हम उनके खाने में मिला देंगे वो खा लेंगे खाना तो वो खाते ही रहते हैं ऑलराइट तो ओरल मेथड इज मोस्टली प्रेफर्ड ओवर इंजेक्शन नाउ कमिंग टू challenges of developing vaccines Now, this is the last slide. And it is important to know कि वैक्सीन क्यों नहीं बन रही भाई हमारी. इट इज़ वेरी इंपॉर्टेंट. और डिजीज़ हमारी इतनी सिवियर है, जान ले लेती है. सिम्टम्स आते ही बहुत ज़्यादा टाइम नहीं रहता है हमारे पास. हमारे मतलब वो इंड्यूस हो जाए बॉडी में वो डिजीज़, तो सिम्टम्स दिखने में भले कई टाइम लगे, लेकिन जब हमारे सिम्टम्स दिख जाते हैं उसके बाद वी डोंट नो कि कब तक वो इंसान अलाइव है. ओके, तो इट्स वेरी इंपॉर्टेंट वैक्सीन हो हमारे पास. ठीक है, लेट्स सी व्हाट आर द चैलेंजेस, क्यों हमारे यहाँ प्रॉब्लम आ रही है. सबसे पहले तो हमारा प्रॉब्लम है कि सेफ्टी रिस्क. सी, देयर आर पॉसिबिलिटी ऑफ प्रियॉन एम्प्लीकेशन. हमें पता है हमारे पास नॉर्मल है, वो कभी भी नॉर्मल में बदल जाएगा. ठीक है, क्योंकि हमारा मिसफोल्डेड प्रोटीन उसके पास टेंडेंसी है टू इन्फेक्ट नॉर्मल प्रोटीन. ठीक है, तो बहुत पॉसिबिलिटी है हमारे अंदर, अगर नॉर्मल प्रोटीन है तो कभी भी किसी को भी अगर एक बार भी प्रियॉन एबनॉर्मल प्रियॉन प्रोटीन से कभी भी सामना हो गया तो वो तो आपको ले डूबेगा. ठीक है, तो इसलिए तो रिस्क है प्रियॉन एम्प्लीफिकेशन. ऑलराइट, the second thing is reactivity to normal prion protein. See, antibody against some regions like, you can say like octa repeats are safe. यहाँ पे specificity की बात हो रही है. तो देखा गया है according to papers there are different regions that the target, you can say that target area, the target region that shows different results. See, antibody against the some regions like abhi I have discussed, I have said a word octa repeats. So octa repeats पे अगर वो target करता है it is safe. But on the other hand, if the antibody targets on for the folded region of the normal prion protein, it becomes harmful to the host, to the body itself. Okay, and it causes neurotoxicity. So it is important कि हमारी ये target completely relies on the specificity of the region. Now the third challenge we face is reactivity to the abnormal prion protein. Now, reactivity to the abnormal prion protein, we all know that abnormal prion protein infect normal prion protein, and it gets, it forms, it aggregates and accumulates later and creates fibers and then, cause neurodegenerative disease. There is no pause. ठीक है, तो सीक्वेंस बनता जाता है, उनकी क्या साइकिल कंप्लीट होती जाती है, साइकिल और साइकिल ही रहती है, उनकी साइकिल खत्म ही नहीं होती है. तो ये हमारा ये हमें पता है कि इट प्रमोट्स एंड स्टेबलाइज द मिसफोल्डिंग ऑफ intermediates. And the second reason is we कि wild types and mutant PRPSCs में different reactions होते हैं एंटीबॉडी के. ओके, नाउ वाइल्ड टाइप्स यानी जो हमारे बाहर से आए हैं इन्फेक्शंस, एंड म्यूटेंट जो हमारे अंदर ही डेवलप हो गए हैं म्यूटेट होकर. ठीक है, नाउ द मोस्ट इम्पॉर्टेंट चैलेंज यू कैन से is lack of knowledge. हमने earlier what we have discussed, the disease is not very old. Okay, so there are not so many researches on this prion disease. Okay, so there is no information on which, on which immune response protects against the prion disease. For example, let us see, we have humoral immune response and we have cellular immune response. Okay, so we don't know that which is important and which will better protects against prion disease. And within these, like within humoral and cellular immune response, we don't know either mucosal immunity works better or either or that peripheral immunity is efficient. So there is so many lack, you can say research gaps. So we don't know what to do, what not to do that will not kill the normal proteins of the human, that humans dies because of lack of different proteins. ठीक है, so this is the reason. So आई होप आपको ये टॉपिक समझ में आया हो. Let's end here. If you have any query, you can contact on this helpline number. Thank you. Have a great day. All right.